Hello, everybody. Welcome back to the second part of the narrated PowerPoints on the digestive system. Hope all of you are doing well, hanging in there. This is a long digestive system. There's going to be part three once we're done with this part two. So let's look back first what we have already looked at, explored in part one of the digestive system lecture. We have looked at workshop number one, the oral cavity. Salivary amylase is digesting starch in there. And we looked at the reflex we call deglutition, swallowing. This pulp of chewed up and wet paste of food is pushed through the esophagus using the motion peristalsis into the stomach. And then in the stomach, you can see the stomach peeking out right here in the top right hand. In the stomach, we looked at gross and microscopic anatomical structures. The mucosa of the stomach is creating hydrochloric acid that is turning the gastric juice and the content of the stomach very acidic, pH 2 or so. The mucosa of the stomach also created those digestive enzymes, pepsin, gastric lipase. They love this acidic pH. They have a field day degrading proteins and triglycerides. We looked at the three-layered smooth muscle of the wall of the stomach, and one function of that set of smooth muscles is to empty the stomach, and this is what this black arrow represents. 1 teaspoonful at a time. This highly acidic chyme from the stomach is being emptied into the duodenum portion of the small intestine. So while the small intestine, especially duodenum, creates a thick layer of mucus that is protecting the first few inches of the small intestine from this acidic chyme. The rest of the small intestine doesn't have that kind of protection, and so we have to prevent the digestion, the degradation of small intestine, and we have to neutralize this pH here. So in this portion of the digestive system lectures, before we move into the small intestine, we need to talk about these two accessory structures, the liver and pancreas. These exocrine glands will be creating secretions, the bile from the liver, pancreatic juice from the pancreas, and the alkali pH of that secretion or those secretions will be mixed with the acetic acid, and it will be neutralizing the acidic pH. This enzyme not only spares the small intestine from damage, but neutral pH is great for the digestive enzymes coming from pancreas, as we'll see later on. Those enzymes are the major digestive factors in the small intestine. So the alkali agent that carries out the neutralization is bicarbonate from the pancreas and bile salts from the liver. The mixing happens early in the duodenum, and after that the neutralized acidic chyme can go on to be digested further and eventually go through absorption in the small intestine. So we'll be starting the track in part two with pancreas and liver. Here's the pancreas. You can kind of see this C-shaped or J-shaped curved shape of the pancreas. The stomach is kind of cut off. The stomach would be emptying into the duodenum right here, and so the pancreas is behind the stomach, underneath the liver. We have seen the pancreas through its pancreatic islets, the pancreas is producing insulin and glucagon. Those are endocrine functions. Most of the pancreas fulfills this exocrine function, producing pancreatic juice, digestive enzymes, plus bicarbonate, and we mentioned this as an agent of neutralization that will help neutralize the acidic chyme from the stomach. We're going to list these digestive enzymes here. They will be active at neutral pH, and these enzymes are digesting anything that the stomach or the salivary stomach enzymes and the salivary amylase did not complete. So we have undigested starch left over. The pancreatic amylase will be degrading that to glucose. We have partially digested proteins coming out from the stomach. The trypsin, chymotrypsin, carboxypeptidase enzymes of the pancreas will be digesting those leftover proteins down to their amino acids. And we also have undigested or partially digested fats and oils, so those triglycerides will be digested by the pancreatic lipase. For the sake of completion, I put here these sets of enzymes, ribonucleases and deoxyribonucleases. From a nutritional point of view, these are not important enzymes. So whenever we consume animal or plant material, if you eat a carrot, you're eating cells of the carrot, and the carrot cells have DNA and RNA in it. When we eat a piece of steak, we have cow DNA and cow RNA in that because we're eating the muscle cells. So what happens to all that DNA and RNA that we are ingesting? Well, in our small intestine, thanks to the pancreatic enzymes ribonuclease, RNA will be degraded and DNA will be degraded by deoxyribonuclease. The degradation products are absorbed but changed in the liver and detoxified, and we are peeing out a good part of the degradation products. We do not use the nucleic acids that we consume to make our own DNA and RNA. Of course, we build our own polysaccharides with glucose that is coming in from animals or plants. We are building our own proteins when we are digesting animal or plant protein. This is not the case for the DNA and RNA. They are just degraded. Consumption of high amount of DNA, usually this happens in people who eat huge amounts of meat. The detoxifying and degrading enzymes can have a mutation, and people carrying mutation where DNA degradation is not complete, those people develop gout. So that's the side effect of not being able to degrade DNA completely in the body. So the pancreatic juice has another important ingredient, sodium bicarbonate. So in addition to these enzymes, we have this alkali agent, and this alkali pH is enough to neutralize the acid in the stomach. It's not written out here, but we are making half a gallon of pancreatic juice every day. So there is a huge volume of this pancreatic juice enzyme and the neutralizing agent ready to take the acidic edge off of that stomach acid. This is how pancreatic juice gets into the duodenum. There is a duct from this exocrine enzyme, the pancreas, the pancreatic duct. We have seen this pattern with the salivary gland. We did not name the anatomical name for that duct, but we have pancreatic duct taking pancreatic juice to the duodenum from the pancreas. For something very different, the liver. So the liver is doing quite a few functions in the body. We are going to start out by looking at the role of the liver in the digestive system, but then we're going to branch out and take a look at other functions of the liver. If you remember, we had liver coming up in the blood chapter, where we use the macrophages in the liver to recycle worn out red blood cells. We also have seen the role of the liver in the blood chapter. We stated that clotting factors, albumin, protein are all made by the liver, and we have seen the role of albumin driving reabsorption in the capillary bed. That albumin is made by the liver as well. So those were just indications that the liver has a number of functions besides making bile. This is the relevant function to the digestive system. So in the first part, I had a brief description of visceral peritoneum. The visceral peritoneum that wraps the liver comes to a sheet-like shape. This is called a falciform ligament. As you can see, the falciform ligament anchors the liver to the diaphragm, and this is one of the example of the anchoring function of these visceral peritoneal membrane. We're not going to say a whole lot about the gallbladder. This is storing bile, the secretion of the liver. So bioproduction. Most of the cells that you would see in the liver, and we'll have a slide about a microscopic structure of the liver, the cells are called liver cells or hepatocytes. They create bile, and there are bile canaliculi and bile ducts that take that bile all the way to the duodenum. So the bile does not contain any digestive enzymes, very different from the pancreas. Excuse me. So why do we prize bile for the digestive system? The key word is right here. It's producing an emulsifier. The emulsifiers are these bile salts chemically, and we'll take a look at in a minute just how emulsification is helped with fat digestion. The bile has a large amount of cholesterol. This is one way our liver disposes surplus cholesterol from a body. Calcium salts of cholesterol in the bile can lead to crystal formation, and gallstones are cholesterol and calcium crystals. We also have coloration of the bile. Pancreatic juice is a colorless, it looks like saliva. The bile has this mean green color to it. Bilirubin is giving the color to the bile, and we looked at already bilirubin way back in the blood chapter. Degraded heme is turned into bilirubin by the macrophages of the liver. So let's take a look at this emulsification. How is it possible that without any digestive enzymes we still use this component bile salts from the liver? So here's the process of emulsification. If you are an enzyme, gastric lipase, and you start digesting a large lipid droplet, it takes you forever to go from the edge and keep digesting it until you get to the other side of this droplet. So to the rescue come biosalts from a liver. So biosalts with the bio are emptied into the duodenum and mixed with the chyme coming from the stomach. And this is what biosalts do to these large lipid droplets. Break them up into many small emulsified lipid droplets. So if you're a pancreatic lipase, you have just a short distance to chew through this small lipid droplet. You are done with a lipid droplet very quickly, and you can go and work on another, a third, and a fourth. So emulsification speeds up enzyme activity by creating a large surface area that allows pancreatic lipase to digest very quickly these small droplets. We use dishwasher, laundry detergents for cleaning our dishes and clothes. Well, very similar action happens on the surface of our clothes or surface of our dishes. Those detergents are emulsifying lipid droplets, whether it's grime from our skin or grease from the leftover dinner. And in our dishwasher, in our laundry machine, we can flush away with hot water the emulsified tiny lipid droplets. And this is how we clean our clothes. This is how we clean better our dishes. So no enzyme action. This is a physical action of the biosalt, breaking up large lipid droplets into thousands of small ones. So here is a running list of other functions of the liver. We'll be looking at this in chapter 25 as well, but liver is a major agent that responds to both insulin and glucagon function. So helping to maintain normal blood glucose, liver is a tool. It's not producing hormones, but it is acted upon by both of those hormones. If blood glucose is low, the liver will be using the stored glycogen, proteins and triglycerides, and we'll see how you can turn protein into glucose in chapter 25. But under the command of glucagon, glucose production starts from the liver. And if we have surplus blood glucose, the liver is going to take that excess glucose and store it away in glycogen and turn it into triglycerides. So whether under the command of glucagon or under the command of insulin, the liver participates at both ends of maintaining normal blood glucose levels. For lipid metabolism, the liver, the hepatocytes make cholesterol. This chemical is tremendously important for cells to stabilize cell membranes. So cholesterol is distributed throughout the entire body. To distribute this lipid and prevent droplet formation in the blood, we package cholesterol and other lipids too into carriers. We just mentioned two here. In chapter 25, we'll mention a number of others. You may have heard these names, good and bad cholesterol, where here's your bad cholesterol, LDL, and here's your good cholesterol, HDL. The abbreviation stands for high density lipoprotein or low density lipoprotein. They are transporting cholesterol. So these are little transporting trucks that move cholesterol around in the bloodstream and either delivers cholesterol to cells or move cholesterol away from the cells. The liver can also store some triglycerides. When the liver stores are full, the liver sends surplus triglycerides to adipose tissue. Taking a look at the protein metabolism, what liver can do with regards to proteins. So the liver can change amino acids as they are coming to the liver. Deamination is a common chemical reaction that is carried out by the liver. The ammonia needs to be turned into urea, and urea is removed through our kidneys, major component in our urine. This is how surplus amino groups or ammonia is removed. A large amount of them is turned into urea. This conversion of ammonia to urea is happening in the liver. And we mentioned already protein synthesis of clotting factor, and the immune system. This reference is to the set of proteins that make up the complement system. We just barely mentioned that in our innate immune system. And because HDL and LDL have a good deal of protein content, so we can list HDL and LDL synthesis as protein synthesis functions in the liver. And just when you thought we were done with the functions of the liver, here is an incomplete list. Liver is able to remove many small organic chemicals from the blood. Detoxification is a set of chemical reactions where those drugs, medications, our own hormones, or any organic chemical that comes into our body will be changed chemically, either put into the bile or released into the blood, and our kidneys can remove those now detoxified chemicals from our body. Producing biosalts from the degraded heme is one function here. We mentioned this already. As the liver can store fat, it can store fat-soluble vitamins as well. The liver can store metals. One detoxifying function of the liver is to take out mercury, lead, cadmium, but the liver also stores metal ions that are needed by our body, such as iron and copper. We have mentioned in the blood chapter this function, removing red blood cells. Those macrophages can also remove bacteria. And bilirubin production is linked to heme degradation from those worn out red blood cells. And the liver is also making or participating in making vitamin D. The process starts in a kidney. The second step is done in the liver, and our kidneys complete vitamin D production. So this is the microscopic structure of the liver. Within the lobes of the liver, and in the lab we'll be mentioning several lobes. Within each lobe there are small clusters or lobules of hepatocytes, and you can see the incoming blood stream, and the blood is flowing between rows of hepatocytes, and you have to zoom in to look at the bile canaliculi merging into bile duct. Bile is released on the opposite end of the hepatocytes in the lobules. This is already an indication of 3 sets of blood vessels participating in liver function. In the lab, we have mentioned both hepatic artery, the beginning of the hepatic vein. What we also mentioned in the blood vessel lab, that there is a vein, the hepatic portal vein, that also brings blood to the liver, and so the flow from the hepatic portal vein and hepatic artery merges, as you can see at this point, and so the hepatic vein is going to drain the liver from the mixture of the oxygenated and deoxygenated blood. So this is the schematic of the blood flow. We have mentioned this term sinusoids way back in the blood vessel chapter. Sinusoids are capillaries that have like Swiss cheese appearing sidewalls. So blood flowing through the liver sinusoid is a mixture of blood from hepatic artery and blood from hepatic portal vein. Hepatocytes and macrophages do their job sitting alongside the sinusoids, and the blood is drained out from the liver first through the central vein, but then to hepatic vein and eventually into the inferior vena cava. How does bile reach the duodenum is a little more complicated than the pancreatic juice. So from lobes of the liver, we have hepatic ducts. They merge into a common hepatic duct. Into the gallbladder, there is a cystic duct that takes stored bile into the common bile duct, and it is this common bile duct that is going to merge with the duodenum and release bile into the duodenum. So with the pancreas and liver fulfilling their function of neutralizing the gastric chyme, adding enzymes from the pancreas, adding an emulsifier from the bile to this mixture of liquid, the flow of the liquid continues through the small intestine. And you can see the beginning of the duodenum here, and the duodenum takes a big turn and eventually continues into the jejunum and eventually into the ileum. And here you can see the coursing of the ileum merging with the large intestine, the cecum portion of the large intestine. So this is the small intestine, duodenum, jejunum, and ileum combined makes up the longest part of the digestive system, huge tube, and we'll be seeing that there will be two tools to increase surface area. Major function of the small intestine is absorption. Without the small intestine, we would be able to absorb very little through the stomach and very little through the large intestine. So yes, chemical digestion is still happening in the small intestine, but the irreplaceable function is absorption. Large surface area will be tremendously helpful. So let's take a look at least the first tool of increasing surface area. So in between these finger-like projections, the intestinal villi is where this intestinal juice and the nutrient-rich, completely digested mix of glucose, amino acids, fatty acids are flowing. And these villi look like the stalagmites and stalactites that come from the sides, the ceiling and floor of the limestone caves. They protrude towards the middle of the small intestine, and they hugely increase the surface area. Another tool to increase the surface area is the microvilli on top of the absorptive cells, and we will have a little more detailed look at that. Goblet cells create mucus. We'll see in part 3 of the digestive system that hormones made by the small intestine, namely secretin and cholecystokinin, is an important part of communication with the stomach. I want to come back to the absorptive cell just for 1 minute. So the presence of villi and the presence of microvilli create a surface area that corresponds to over 2,000 square feet in our small intestine. If you live in a house or an apartment, you know the area of that. My home is a lot smaller than 2,000 square feet, and so when you look around in your house, your small intestine has a larger surface area probably than the entire living area of your house. And it's all thanks to the villi and the microvilli increasing surface area. The microvilli, besides increasing surface area for absorption, also adds a new set of enzymes, and I believe my artistic rendition of this is depicted here. So here's an absorptive cell with the microvilli, these black spikes here, and if we take a closer look, we would see digestive enzymes not swimming in the chyme over the absorptive cell, but some digestive enzymes are stuck on the surface. These red X's represent digestive enzymes. So they sit tight, and they are removed only when the absorptive cell is ruptured as the material is moving through the small intestine. So what do these enzymes add to the digestion, in addition to the pancreatic enzymes that are carrying out the degradation in the lumen of the small intestine? So look at these two names here, lactase and sucrase. We consume a large amount of chemicals that need very, very little degradation. For example, sucrose is the cane sugar that we put in pastry, coffee, tea, and that sucrose is a disaccharide. All it needs is one cleavage between the glucose and fructose components. Sucrase enzyme carries it out here. This is the only place where cane sugar is degraded in our body. Without the degradation, the absorptive cell cannot absorb cane sugar. Lactase is the enzyme that degrades milk sugar. So milk sugar, lactose, needs to be degraded by lactase before the two components, glucose and galactose, can be absorbed. And so lactase enzyme carries that out. Lactose intolerance is coming from the mutation or complete lack of this enzyme, and undigested lactose, milk sugar, will go into the large intestine, and bacterial digestion will cause the bloating, the diarrhea, and gas production, the symptoms of lactose intolerance. These red X's together are called brush border enzymes because under the microscope these microvilli look like a brush bordering the absorptive cells. So the summary of the digestion types in the small intestine: we do have peristalsis, And this image here represents peristalsis. You see the contraction of the smooth muscle in the muscularis of the small intestine, and this contraction is moved along, helped by the enteric nervous system, and the muscle contraction is coordinated to keep moving along this bolus, along this 20-foot long small intestine. We also have segmentation. Segmentation is showing here. Segmentation is a mixing type of movement where low and high concentration of materials can be evened out by periodic contractions of the walls of the small intestine. When it comes to chemical digestion, we have to refer back to those pancreatic enzymes, the pancreatic lipase, pancreatic amylase, trypsin, chymotrypsin. These enzymes act in the lumen of the small intestine. This is the material that is swimming in between the intestinal villi. And then we have the brush border enzymes, degrades carbohydrates. Some of those enzymes degrades peptides into amino acid. So this is the other set of enzymes contributing to chemical digestion in the small intestine. So our last big topic here is absorption. So we have seen the importance of movement of molecules in and out of the bloodstream in the respiratory system. We must somehow move oxygen into blood. We have seen the respiratory membrane promoting the diffusion of oxygen. In the case of the small intestine, absorption is the movement of the digestion products into the blood. So instead of oxygen, now we're talking about glucose, amino acids, fructose, fatty acids, and eventually we'll be mentioning things like vitamins, electrolyte, and water. So to make this movement a little easier, our small intestine creates about half a gallon of intestinal juice that is going to create a water flow that will move many materials into the blood. In addition to diffusion, we will see the role of facilitated diffusion, active transports. Active transports always use ATP, that's their hallmark. And then we also see movement of water, and this is the only way to describe movement of water: osmosis. So here's the schematics of our small intestine. You see 3 absorptive cells here in yellow. Here's the brush border microvilli, and here's the bottom of the cells facing the blood vessels. So let's see what happens to all these nutrients in the lumen of the small intestine. Glucose and galactose. Those sugars are absorbed through active transport, secondary active transport together with sodium ions. Secondary active transport is where sodium ions first pumped out of the cell into the lumen, and then sodium ions streaming back into the cell will drag with them glucose and galactose. We have a very similar transport mechanism for amino acids. Dipeptides and tripeptides can co-transport with hydrogen ions. So these are all active transports, require ATP at the very beginning. Fructose, and this is a monosaccharide found frequently in fruits and vegetables, honey. Fructose enters the cells using facilitated diffusion. Once these materials enter the cell, they lead to a high concentration inside the cell, and usually concentration-dependent mechanism, facilitated diffusion or diffusion, moves them into interstitial fluid and eventually into the blood. So this is the absorption of simple sugars, amino acids, dipeptides. We're going to have two separate slides for looking at fatty acid absorption. Short-chain fatty acids just enter the absorptive cell through simple diffusion, but the fatty acids that come from the triglycerides have a very, very different fate. As you can see, the bracket closes here, so we are moving the absorbed fatty acids into something else, not into the bloodstream. So let's take a look at the first part of the lipid absorption. So we created fatty acids, even some monoglycerides, and these molecules enter the absorptive cells by simple diffusion. What happens inside the cell is totally unprecedented because the absorptive cell takes fatty acids, monoglycerides, or glycerol, and rebuilds, reconstructs triglycerides right here inside the absorptive cell, packages them into huge beach ball shaped structures called chylomicrons, and toss the chylomicron out of the cell. So once the chylomicrons are outside the cell in the interstitial fluid, they are too large to enter into bloodstream. So with the passive flow of the lymph, the lymphatic capillary or lacteole of the small intestine will absorb the chylomicrons, and with the loaded triglycerides and the fat-soluble vitamins. So the freshly absorbed triglycerides in the chylomicron go on a different trip through the lymphatic system, while everything else was absorbed into the bloodstream. Let's take a look at the absorption of electrolytes. So these are chemicals that we consume: sodium, potassium, chloride, calcium. They don't need to be digested by enzymes because as soon as we swallow them, they are ready to be absorbed. So sodium and potassium are absorbed through active transport, through the contribution of sodium and potassium pumps. Chloride, iodide, we've seen iodide needed for thyroxine production. They all, and nitrate, enter absorptive cells by diffusion. Iron, magnesium, phosphate, and calcium, they require active transport, and so we are spending ATP to move iron, magnesium, phosphate, and calcium into the absorptive cell. What about vitamins? So the fat-soluble vitamins is really easy. They go with the the ingested lipids. They are going to be absorbed into the chylomicron, and they will be moving along with those triglycerides. Water-soluble vitamins are absorbed by diffusion. There's one notable exception here, which is vitamin B12. Vitamin B12 needs a protein intrinsic factor. Without that, vitamin B12 cannot be absorbed. Vitamin B12 deficiency sometimes can be corrected only by injection. If the patient's intrinsic factor is not there, orally taken vitamin B12 will be no good. It will not be absorbed without intrinsic factor. And at last, let's take a look at water. In chapter 26-27, we'll be looking at the importance of water. We do have water homeostasis in our body, and so this is the way we absorb water. The type of movement is osmosis. And in addition to what we are consuming, 2-3 liters, that's about a little more than half a gallon. But look at how much more water our digestive system needs to handle, especially the small intestine. We make about 2 water bottles worth of saliva every day. We make 2 water bottles worth of 1 liter of bile every day. This is all going into our digestive system, right? We make half a gallon of gastric juice. I already mentioned we make half a gallon of pancreatic juice. We make 2 water bottles worth of intestinal juice. So altogether, we're looking at over 2 gallons of almost 3 gallons of liquid that needs to be absorbed. Most of the water is absorbed by osmosis in the small intestine, and about 10% or so absorbed in our large intestine. So in addition to absorbing organics, electrolytes, vitamins, water absorption is also happening in our small intestine. Water again moves by osmosis, absorbed by osmosis. And to close this lecture, let's take a quick look where everything is heading to. So material absorbed into the bloodstream is going to the liver through the hepatic portal vein. In the lab, we mentioned that superior mesenteric vein, together with gastric, splenic, and inferior mesenteric veins, are all merging into the hepatic portal vein. What about those triglycerides and fat-soluble vitamins? They absorb into the lacteal lymphatic capillary, and they flow with the lymph all the way to the subclavian vein, and then the subclavian vein will be carrying the freshly absorbed lipids in chylomicrons, and eventually those chylomicrons will reach the liver as well. So I'm going to close part 2 here, and we have a much shorter part 3 to conclude the digestive system.